I want to start with a story that honestly kept me up last night. It's from the literature about a child they call Bella.
Hmm.
And I want you to just picture this. It's the night before sixth grade.
Okay. High stress situation already.
Totally. I mean, you're already worried about your locker, your new classes, who you're going to sit with at lunch, all that stuff. But for Bella, her parents sit her down for a conversation that is, I mean, it's about as heavy as a conversation could possibly get.
It's the kind of talk that doesn't just change your future, it completely rewrites your past.
Exactly. This isn't just, a family secret. This is a fundamental biological reality that's been hidden for 12 years.
Yeah.
They tell Bella that when she was born, she wasn't a girl. Genetically, she was male. She had tests. But, and this is the core of it, she was born with a condition called cloacal exstrophy. It's incredibly rare.
Oh, extremely rare. We're talking maybe one in every 400,000 birth.
Yeah.
It's a developmental issue where the abdominal wall just doesn't close properly.
So the skin that's supposed to cover the lower abdomen, the stuff that forms the external genitalia,
Mm,
it's just missing.
right. So in this case, for Bella, even though the child was genetically male, XY chromosomes, testes, there was no penis.
And this is where the story just stops being a medical case for me and it becomes this huge ethical, biological dilemma.
Absolutely. Put yourself in the parent's shoes.
Yeah. You have a baby boy, but he has no penis, so what do? Do you raise him as a boy, knowing he's going to face, I mean, just an incredible number of surgeries, potential psychological trauma around his identity, his sexual function, or do you take the other path?
And you have to remember the context of the time. This wasn't happening in a vacuum. The prevailing medical wisdom, the sort of dominant theory then, was that gender identity was mostly learned,
A blank slate?
a blank slate, nurture over nature. So the recommendation and the decision that was made was to intervene surgically. They removed the testes, they constructed genitalia that looks female,
Yeah.
and the advice to the parents was very clear.
Raise this child as a daughter.
Put her in dresses, give her dolls, call her she, treat her as a girl in every single way, and she will be a girl. The identity will follow the upbringing.
And they did. For 12 years, Bella lived as a girl, socially, legally, in every way. But then comes that night before sixth grade. The parents finally lay it all out. You were born male.
Hmm.
And this is the moment that just it floors me. You'd expect what? Shock, denial, tears.
Hysteria, maybe? A total identity crisis?
Right. But Bella didn't cry, didn't panic, just looked at them and said, I knew it was true.
I knew it was true.
Can you imagine?
It's a chilling line. She apparently went on to say something like, even if another person had told me, I would have believed them because it just made sense to me.
It made sense. And the
Yeah.
fallout from this, the transition, is even more stunning. This 12-year-old socializes a girl for over a decade immediately. And I mean, like, instantly reverted to a male identity.
He took the name Benjamin.
Right. And later, when doctors asked him, how did you do that so fast? How did you change your entire life, your whole identity in a week? His answer was just, it was so simple. He said, cut my hair. That's all I did.
It's just a staggering case study. It really is. And it forces you to confront the limits of, how we think about ourselves.
Right.
We love this idea that we're these blank slates, that our culture and our parents write the script.
But Benjamin's story, it just suggests there's something else, something hard-coded, something writing the script from the inside out long before we're even aware of it.
That's the feeling, that
Yeah.
we are, in a sense, passengers in this biological machine that has its own directives, its own drives, its own sense of identity, regardless of what we try to paint on the outside.
And that's really the core theme of what we're getting into today. We're doing a deep dive into the biological machinery that runs our most basic, most primal behaviors.
We feel like we're the pilot, making all these rational choices. But when you dig into the behavioral neuroscience, especially following the path of Breedlove and Watson's text, you realize the ship is very often on autopilot.
We're talking about the absolute bedrock hardware that dictates survival, and we're going to break it down into three big areas.
Yeah.
First, we'll tackle the machinery of sex, not just the physical act, but the deep evolutionary programming of attraction, differentiation, everything that goes into continuing the species.
The why and the how.
Exactly. Then second, we'll look at homeostasis. This is the body's obsession with staying the same, the relentless, redundant systems that keep us fed and watered and our internal environment stable.
Keeping the machine running.
Keeping the machine running. And finally, we're going to talk about the clocks, the invisible timers inside our cells that decide when we wake up, when we fall asleep, and, arguably when we are even conscious.
So reproduction, regulation, and rhythm.
That's it. So let's start with what is maybe the most fundamental drive of all, making more of us.
Right. The text from Breedlove and Watson breaks reproductive behavior down into 4 distinct stages, which I found really helpful because culturally we just lump it all together and call it sex, but biologically it's a very specific sequence of events.
It's a cascade, yeah.
Yeah.
One stage has to happen for the next one to be possible, and it starts long, long before any kind of physical contact. It begins with stage one, sexual attraction.
Which of course brings us to the peacock. You can't talk about attraction in biology without talking about that tail.
It is the absolute classic example, and it's perfect because it illustrates this tension between sexual selection and, well, natural selection.
What do you mean by that?
Well, think about it just from a pure survival point of view. A peacock's tail is a disaster. It's heavy, it's cumbersome, it makes it hard to fly. It's also incredibly bright and flashy. It's like wearing a giant neon sign that says, "Hey, predators over here, lunch is served."
Okay, so from a camouflage and escape perspective, it's a terrible idea. A terrible idea.
Yeah.
So the question is, why would evolution allow it to persist? Why haven't the peacocks with the smaller, duller, more practical tails won out?
Because the peahens, the females, they're the ones doing the choosing. And they like the big tail?
I love the big tail, but why? That's the deeper question. Why would a female actively choose a mate who is, on the surface, less likely to survive? Is it just, an aesthetic preference? Ooh, that one's sparkly.
I'm guessing it's more than that.
It is. It goes back to this idea called the handicap principle. The tail isn't just an ornament, it's an honest signal of genetic quality.
An honest signal.
Yeah, the male is essentially advertising, look at me. I am so healthy, so strong, so genetically fit that I can grow this ridiculous, burdensome tail and still be alive. I am surviving despite this handicap.
So it's a flex. It's the ultimate flex.
It's like a billionaire lighting a cigar with a $100 bill just to show they can afford to waste it. That's a perfect analogy. It signals excess capacity. It says, my genes are so good I can afford to waste resources on this beautiful, useless thing. So the female isn't choosing the tail itself. She's choosing the underlying genetic vitality that the tail represents.
Wow. Okay. So that's attraction. Now, what about moving from just attraction to, relationships? The text spends a lot of time on voles.
Yes, the vole.
I always love the vole studies. They seem to be the go-to animal model for studying monogamy.
They are. And it's because you have these two closely related species with dramatically different social structures. You have meadow voles, which are highly promiscuous, and then you have prairie voles, which form these very tight long-term pair bonds.
They look like the perfect little nuclear family, right? They live
Yeah.
together, they raise the pups together, they defend the nest.
On the surface, yes. But this is where the text makes a really sharp and important distinction. There's social monogamy, and then there's sexual monogamy.
And they are not the same thing.
Not at all. Social monogamy is about the partnership. Who do you live with? Who do you build a nest with? Who helps you raise the kids?
Okay, the domestic arrangement.
Right. Sexual monogamy is about who you actually meet with. And it turns out that even our faithful little prairie voles are pretty frequently engaging in what the researchers politely call extra-pair copulation.
Which is the scientific way of saying they're stepping out on their partner.
It is. And this creates a fascinating evolutionary puzzle. Why do it? If you have a stable pair bond, why risk the energy, the potential conflict?
Well, from the male's perspective, it seems pretty straightforward. It's the old, drive to spread your genes as widely as possible. Low investment, potentially high reward.
Exactly. Classic male reproductive strategy.
Mhm.
But the females do it too. And for the female, the calculation is a bit more subtle, a bit more complex.
What's in it for her?
Genetic diversity for her litter. Think about it. By having a secret rendezvous with a male from outside the pair bond, she's essentially hedging her bets.
Like an insurance policy.
Precisely. Her main partner might be a great dad, a great provider, but what if he has some hidden genetic flaw? The extra pair encounter ensures that she isn't putting all of her genetic eggs in one basket.
That is coldly calculated.
That is.
It completely decouples the social, maybe even emotional bond from the raw biological act of reproduction. Like
Sure.
the vole is saying, I love you, you're a wonderful partner, but I need to diversify my genetic portfolio.
That is a fantastic way to put it. Nature prioritizes the outcome, which is healthy, diverse offspring over any sentiment of, total exclusivity.
Okay, so we've got attraction, we've talked about relationships. Moving down the list, the text has stage 2 as appetitive behavior.
Aptive behavior. This is all the stuff that happens to establish, maintain, or promote the sexual interaction.
So if attraction is the spark, this is fanning the flame.
Exactly. This is the advertising phase. In many species, this is known as a perceptive state, where the female in particular advertises her readiness to mate.
And the text gives the example of ear wiggling in female rats, which I have to say sounds kind of adorable.
It is a very distinct behavior. A female rat who is receptive doesn't just sit there and wait. She'll approach the male, then dart away, she'll hop, and she'll wiggle her ears. It's a specific, coded signal that triggers the male's interest.
But in primates, the signaling can be much more physiological, less behavioral.
Oh, much more. You get the phenomenon known as sex skin.
Right.
In many species of macaques and baboons, this advertising is perfectly synchronized with the female's internal hormonal clock. So when she's ovulating, when her estrogen levels are peaking, and conception is most likely.
The skin around her genitals swells up and turns bright red or pink.
Exactly. The body basically turns on a giant flashing billboard that says, "Open for business."
And this ties right back into our core theme of the machinery. The behavior, the advertising, isn't just a whim or a mood. It's locked in step with the endocrine system. The brain, the ovaries, the skin, they are all coordinating to make sure that sex happens at the precise moment it can lead to reproduction.
A beautifully synchronized system.
Which brings us, logically, to stage 3, copulation itself, the mechanics.
Intermission and ejaculation. But what I found most interesting here in the
Right.
text's discussion was the concept of the Coolidge effect and the refractory period.
Let's break that down, because the Coolidge effect sounds like a political thriller, but it's definitely not.
Yeah, no, not at all. It's named after a probably apocryphal story about President Calvin Coolidge visiting a farm, but the biological principle is very real.
What is it?
It refers to the observation that an animal, typically a male, who has mated to the point of exhaustion and refuses to mate any further with the same female.
He's done.
He's done. But if you suddenly introduce a new receptive female, he will revive and mate enthusiastically all over again.
So the exhaustion isn't purely physical, it's more like sexual habituation, boredom.
It's a refractory period, a downtime, that can be overridden by novelty. But, and this is important, there is a limit. The text is very clear about the absolute refractory period in human males. After ejaculation, there's a physiological window of time where a male simply cannot become erect or ejaculate again. The machinery needs to reset. The plumbing needs to recharge.
But this is where the text contrasts the male and female response, and they use those famous charts from Masters and Johnson back in the 1960s.
Right, figure 12.9 in the text. And this highlights a key difference in the hardware. The typical male response, as they mapped it, is very linear. You have 4 phases: excitement, plateau, orgasm, and then resolution, which includes that refractory period. It's a predictable cycle.
But the female response was different.
Much more variable. When they mapped the female response, they didn't find one single pattern. They found at least 3 common ones.
What were they? I remember seeing 3 different lines on that graph.
Exactly. So one pattern did look a lot like the male pattern, a rise to orgasm and then resolution. But another common pattern showed a rise to the plateau phase, staying there for a while without a distinct orgasm, and then a slow resolution. And the third pattern was the one that really stood out. It showed a sharp rise to orgasm, followed almost immediately by another orgasm, and then potentially another.
So multiple orgasms in quick succession.
Yes. The key takeaway being, for females, there is no absolute refractory period.
Which suggests, if you want to think about it in mechanical terms, that the female system is actually capable of a higher throughput, a higher frequency of response than the male system, which is strictly gated by that reset timer.
That's a fair interpretation of the Masters and Johnson data, yes.
Yeah.
The potential capacity is different.
Okay, I want to pivot now. We started this whole thing with Bella with this idea of the body's biology shaping the brain's identity. And I feel like the most mind-bending part of this chapter is where the text details a specific mechanism that shows exactly how this happens.
You're talking about the SNB.
The spinal nucleus of the bulbar cavernosus.
It's a mouthful.
It's a total mouthful. But this mechanism, to me, it's like the smoking gun for how biological sex differences get physically wired into the nervous system.
It really is one of the clearest examples we have. So let's try to visualize this. Figure 12.22 in the book lays it out beautifully.
Go ahead.
Deep in the spinal cord, in the lower back, there's a specific cluster of motor neurons. This is the SNB. And the job of these neurons is to control a particular set of muscles, the bulbocavernosus muscles, which are attached to the base of the penis.
Okay. Makes sense. You need neurons to control those muscles. But here is the kicker, the part that got me. Females have these neurons too, right? At least at the beginning.
That is the crucial point. In the early fetal stage, both males and females have the full set. They have the SNB neurons in the spine, and they have the muscles that those neurons connect to. We all start with the same basic kit.
So what changes?
The genetic programming kicks in. In a male fetus, the newly formed testes start producing a flood of androgens, specifically testosterone.
And that testosterone goes everywhere in the body, including to those muscles at the base of the would-be penis.
Right. It circulates in the blood, and it binds to androgen receptors on those specific muscles. Now, here is the magic step. When testosterone binds to those receptors, it saves the muscle. The muscle survives and thrives.
Okay.
And because the muscle survives, it starts producing and releasing something called trophic factors.
Trophic factors. I always think of these as like a little care packages or fertilizer for neurons.
That is a perfect analogy. The muscle is essentially sending these chemical care packages back up the axon to the neuron in the spine that controls it. And as long as the SNB neurons receive these trophic factors, they stay alive.
So let me get this chain of events straight. Testosterone saves the muscle. The saved muscle then feeds the neuron. The fed neuron lives.
You've got it. That's the male developmental pathway. Now let's look at the female fetus.
No testes.
No testes, so no big testosterone spike early on. This means the androgen receptors on those bulbocavernosus muscles, they sit empty. There's no signal.
And because they're empty.
The muscle gets the message that it's not needed. It shrivels up and dies. It undergoes apoptosis, programmed cell death.
So no muscle means no care packages.
Exactly. The neurons up in the spine are, in a sense, waiting for a delivery that never arrives.
Yeah.
And when they're deprived of those trophic factors from the muscle they were supposed to control, they undergo apoptosis too.
They self-destruct.
They literally disappear.
Yeah.
So by the time the animal is born, the male has this robust, functional cluster of neurons in his spinal cord, and the female has almost none.
That is just, it's wild. It means the difference between a male and a female nervous system, at least in this one spot, isn't just about software or what hormones are floating around later in life. It's about which wires were physically kept and which ones were cut during the initial construction.
It's use it or lose it applied at the cellular level before you're even born. The hormone sculpts the hardware.
And this isn't just happening in the spine, right? I'm thinking about the brain scans, the text mentions, the ones taken during orgasm. It feels connected.
Figure 12.27, yeah. They use fMRI to see what parts of the brain activate during climax. And you see what you'd expect, of course. You see activity in the basal forebrain, part of the reward circuit. The pleasure button gets pushed.
Sure.
But what's really interesting is that you also see widespread activation across the cortex, including heavy activity in the prefrontal cortex.
The PFC, that's the CEO of the brain, right? The part that does abstract thought, planning, self-awareness. The part that says I.
The very same. Which implies that for humans, sexual release isn't just a brainstem reflex. It's not just a physical event. It's deeply entangled with our consciousness, our self-awareness, our whole cognitive apparatus. It's a whole brain event.
Which I guess brings us right back to Benjamin. His powerful sense of male identity wasn't just in his genes or his upbringing. It was likely structured into the very architecture of his brain and spine in ways we're only just beginning to understand.
You can change the external hardware with surgery, but you can't so easily rewire the nervous system that was built to map and run that original hardware.
All right, let's shift gears. A big shift. We've covered the survival of the species. Now let's talk about the survival of the individual. Homeostasis.
The body's relentless drive to keep things the same. Balance.
And the key word the text uses over and over in this chapter is redundancy.
It's the central design philosophy of any life support system. Think about it. If you're an engineer designing the systems for a spaceship, you don't build just one oxygen sensor.
Right. Because if that one sensor fails, everyone dies.
Exactly. You build 2 or 3 or 4 different systems that all monitor the oxygen levels, and they all check each other. The body's the same way. Evolution values survival above all else, so it built in backups for the backups.
Okay, so let's apply that to thirst. I honestly used to think thirst was a pretty simple signal. You know, my mouth is dry, I should drink. But the book explains there are actually 2 totally different kinds of thirst that trigger the same behavior.
Right. There's hypovolemic thirst and osmotic thirst.
Okay, so hypovolemic. Hypo meaning low, volemic meaning volume, so low fluid volume, like if you're bleeding or...
Or you've been sweating profusely on a hot day, or you have severe vomiting or diarrhea. You have physically lost fluid from your body. Your overall blood volume drops, your blood pressure falls, and special sensors called baroreceptors, pressure sensors in your heart and major blood vessels, detect this drop and scream at the brain, danger, pressure is falling, refill the pipes.
That makes perfect sense. It's a mechanical problem. But osmotic thirst seems way more subtle. The book says, this is the bag of pretzels thirst.
Chuckles, that's a great way to describe it. In this case, you haven't lost water, but you've added a ton of solute, in this case, salt, to your bloodstream.
So your blood gets too salty.
Exactly. And basic physics dictates that water will always try to move from an area of low salt concentration to an area of high salt concentration to balance things out. It's called osmosis. So the water inside your body's cells gets sucked out into the bloodstream to try and dilute all that salt you just ate.
Wait, so your cells? They literally shrink.
They shrivel up. They become dehydrated from the inside out. And you have specialized neurons in your brain called osmoreceptors whose entire job is to feel if they themselves are shrinking.
But hold on a second. I thought the brain was protected. The blood-brain barrier, right. It's supposed to be this super tightly controlled gate that keeps things in the blood from just wandering into the brain. How can the brain paste how salty the blood is?
This is one of my absolute favorite details in all of neuroanatomy. There are these special little structures called the circumventricular organs. The text highlights a couple of them, the OVLT and the subfrontal organ.
Sounds like a location from a sci-fi movie.
It kind of is. Think of the blood-brain barrier as a massive wall around a fortress. But in these specific tiny little spots, the wall has windows. The barrier is deliberately leaky or incomplete.
So it's a security loophole on purpose?
A purposeful loophole. It allows the neurons in these circumventricular organs to stick their little chemical tums out and directly sample the composition of the blood as it flows by.
They are literally tasting the blood to see if it's too salty or not.
That's exactly what they're doing.
Yeah.
And if they detect that it's too salty, that the cells are starting to shrink, they trigger a massive brain-wide alert that we experience as the sensation of thirst.
And you can't fool them. I loved the study that text describes, the wet mouth study.
Oh, it's a classic. The researchers wanted to ask, is the feeling of thirst just about the sensation of a dry mouth? So they took very thirsty people, put them in a brain scanner, and just squirted water into their mouths to wet them without letting them actually swallow and drink.
And what happened?
Well, their self-reported feeling of thirst went down a little bit for a few minutes. The brain got the signal, okay, water is near, this is good, but the feeling came right back. And more importantly, the brain scans showed that the deep primal thirst circuits in the limbic system and cingulate cortex, they didn't shut off at all.
They stayed lit up.
They stayed bright red until the person actually drank the water, swallowed it, and the body sensors confirmed that fluid was being absorbed and the blood composition was starting to change.
The brain is basically saying, nice try with the wet sponge, but my internal osmoreceptors are telling me my cells are still shriveling. Drink the water.
The redundancy again. The system checks the final result, not just the initial input.
Okay, so let's move from thirst to hunger. If thirst is like a two-part harmony, hunger feels like a full symphony orchestra where half the instruments are out of tune and fighting with each other.
That's a very good way to put it. It is infinitely more complex. You're not just managing one substance, water. You're managing glucose levels, short-term energy, long-term fat stores, amino acids, vitamins, minerals, and matching it all to your energy expenditure.
And the messengers that coordinate all this are hormones. The text lays out a whole cast of characters. We've got ghrelin, leptin, PYY, insulin. Let's try to clarify who does what. Who's the villain in this story?
Well, if you're on a diet, ghrelin definitely feels like the villain. It's the hunger hormone.
Ghrelin sounds like growling. That's how I remember it.
That works perfectly. Ghrelin is released primarily by the stomach when it's empty. Its levels in your blood spike right before your normal meal times and then drop dramatically right after you eat. It's the short-term signal that screams to the brain, the tank is empty, feed me now.
Okay, so ghrelin is the gas pedal for eating. What about leptin?
Leptin is the opposite. Leptin is the long-term auditor. It's a hormone that's released by your fat cells, your adipose tissue.
So the more body fat you have, the more leptin you have circulating in your blood.
Exactly. And leptin's message to the brain is essentially, hey, we have plenty of energy stored away in the bank. We're good. You can ease up. You don't need to eat as much, and you can burn a bit more energy.
So in theory, that sounds like a perfect self-correcting loop. If I gain weight, my fat cells get bigger, my leptin goes up, my appetite should go down, and I should naturally lose the weight.
In a perfect logical world, yes, that's how it would work. But the system is not neutral. It's heavily biased.
Biased how?
It's biased toward preventing starvation. From an evolutionary perspective, dying of starvation was a much, much bigger threat for millennia than dying of obesity. So the eat more signals are much stronger and harder to ignore than the eat less signals.
Let's look at where these signals all converge. The control center, the arcuate nucleus in the hypothalamus.
This is it. This is the grand central station for energy balance. It's a tiny little cluster of neurons, but it's where all these hormonal messages from the body, leptin from the fat, ghrelin from the gut, insulin from the pancreas, all arrive and are integrated.
And inside this nucleus, you have two opposing teams of neurons, right?
Two teams in a constant battle. You have the POMC neurons and the NPY neurons.
Okay, POMC and NPY.
The POMC neurons are the brake. They are the satiety neurons. When they're active, they make you feel full, they suppress your appetite, and they tell the body to ramp up metabolism and burn energy.
And the NPY neurons.
They are the gas pedal. They are the hunger neurons. When they get activated, they stimulate ravenous eating, and they simultaneously tell the body to conserve energy, to turn down the metabolic rate.
So my body weight is basically the result of this constant arm wrestling match in my hypothalamus between the POMC stop eating cells and the NPY start eating cells.
That's a fantastic summary of it, and this explains precisely why sustained weight loss is so brutally difficult.
Because of that evolutionary bias.
Yes. The moment you go on a diet and start losing weight, your fat cells shrink. That means your leptin levels plummet.
And low leptin is a huge alarm signal to the brain.
It's a 5-alarm fire. Low leptin tells the arcuate nucleus famine has struck. So what does it do? It takes its foot off the POMC brake, and it slams the NPY gas pedal to the floor.
So my own brain starts fighting me.
It fights you tooth and nail. It makes you intensely hungry. It makes food seem more rewarding. And it slows your metabolism to a crawl to conserve every last calorie. Your body is defending its weight because it thinks you're starving.
This makes the discussion in the text about surgical interventions like Roux-en-Y gastric bypass make a lot more sense. It's not just about physically making the stomach smaller.
No, that's only part of the story. The Roux-en-Y procedure, as shown in figure 13.25, creates a tiny stomach pouch and then reroutes the intestine to connect directly to it. Yes, it restricts the volume of food you can eat, but maybe more importantly, it profoundly changes the hormonal profile of the gut.
How so?
It alters the release of hormones like ghrelin and PYY. It essentially hacks the system and changes the signals being sent to the brain, making people feel fuller on less food.
But the part of this chapter that really blew my mind was the section on the microbiome.
The final frontier of gut health.
The book talks about fecal transplants, and I know it has a high yuck factor, but we have to get past that because the implications here are just staggering.
They are paradigm shifting. The core concept is that your gut, particularly your large intestine, is not just you. It's an ecosystem populated by trillions of bacteria. And my ecosystem is different from your ecosystem.
And those little passengers might actually be driving the bus.
There is compelling evidence that they are at least grabbing the steering wheel. The text mentions the classic studies where researchers take gut bacteria from a genetically obese mouse and they transplant it into a lean, germ-free mouse that was raised in a bubble.
So the lean mouse gets a new gut ecosystem. And what happens?
It becomes obese. Even if it's eating the exact same amount of food as a lean control mouse, it gains significantly more weight.
That's terrifying. What's the mechanism? Are the bacteria from the obese mouse just more efficient at extracting every last calorie from the food?
That appears to be a big part of it. If you and I both eat an apple and you have an efficient microbiome, you might absorb 100 calories from it. I, with my less efficient microbiome, might eat the same apple and only absorb 70 calories. The rest passes through.
This completely upends the simple calories in, calories out model we always hear about with obesity. It's not just about willpower or choices if the very ecosystem you're hosting is working against you.
It redefines what self even means in this context. Are you the individual human, or are you the host of a multi-species colony? And it raises an even darker question.
Which is?
Who is actually hungry?
What do you mean by that?
Is it you craving that donut? Or is it a specific population of sugar-loving bacteria in your gut that are thriving, and when their food source gets low, they send chemical signals up the vagus nerve directly to your brainstem, manipulating your cravings to say, hey host, go get us a donut.
Wow. So I'm just a puppet, and my gut bacteria are the puppet masters.
It's a bit of an exaggeration, but it highlights the complexity. The body defends its weight not just because of your own genetics and hormones, but because there's a multi-species alliance working together to maintain the status quo.
Okay. We have fed the machine, we have watered it, we have reproduced. Now the machine needs to rest and recharge. Segment 3, rhythms and sleep.
And this chapter starts, as it always should, with a hamster on a wheel.
The poor, tireless hamster.
Cockles. The hamster is the hero of chronobiology. It's the classic experiment. Hamsters are nocturnal, so they run on their wheels all night and sleep all day. If you put them in a lab with the lights on for 12 hours and off for 12 hours, they sync up perfectly. They start running the minute the lights go out. This is called entrainment.
But the huge question was, is the clock that's driving this behavior internal to the hamster, or is the hamster just reacting to the external light cue?
So to find out, they did the simple but brilliant thing. They just turned the lights off and left them off. Constant dim light for weeks.
And the hamster didn't just start running at random times.
Not at all. It maintained a very consistent rhythm. It would wake up, run for about 12 hours, and sleep. But the whole cycle was just a little bit longer than 24 hours, maybe 24 and a half hours. So each day it started a little later. This is called free running.
And that drift proved that the clock had to be endogenous. It's inside us.
Exactly. The light doesn't create the rhythm. It just resets it each day.
And we now know exactly where that master clock is, the SCN.
The suprachiasmatic nucleus. A tiny, tiny pair of nuclei in the hypothalamus, located right above the optic chiasm, where the optic nerves cross.
But here's the thing that always confused me. If the clock is internal, why do we get jet lag? If my clock is inside me, why should it care if I fly from New York to Paris? Why does light matter at all for resetting it?
Because that internal clock, that free running period, it isn't perfect. For most humans, it's a little bit longer than 24 hours, just like the hamster. So every single morning, it needs to be nudged back a little to stay in sync with the actual rotation of the Earth. It needs to be reset by the sunrise.
And it gets that reset signal from light hitting our eyes. But, and this is a key detail from the text, not from the rods and cones we use to see images with.
No, this is a completely separate non-visual pathway. There's a special class of retinal ganglion cells in your eye that contain a unique photopigment called melanopsin. These cells aren't for seeing shapes or colors. Their only job is to detect the overall ambient brightness level.
So a person who is blind, who can't see the world visually, might still have these cells working perfectly fine.
Exactly. And if they do, their body still knows when it's daytime and when it's nighttime. These melanopsin cells have a direct line, a neural pathway called the retinohypothalamic tract that goes straight to the SCN. And every morning when they detect light, they send a signal to the SCN that says, sun's up, reset the clocks.
Okay, now we have to get into the weeds a bit. The text describes the molecular clock, the actual ticking mechanism inside the individual SCN cells. It's this loop of proteins. Clock, cycle, purr, and cry.
It sounds like a strange law firm.
Yeah,
Clock, cycle, purr, and cry.
tuckles. It really does. But let's try to simplify this mechanism. How does a single cell tell 24-hour time?
The book uses a great analogy. Think of it like a heater in a room that's controlled by a thermostat.
Okay, heater and thermostat, I can picture that.
The heater is made of two proteins, clock and cycle. They bind together, and then they bind to the cell's DNA. And when they do, they act as transcription factors. They turn on the production of other genes. They start heating up the room.
And the heat in this analogy is?
The heat is the production of two other proteins, purr and cry. So the clock cycle heater is running, and the cell starts to fill up with more and more purr and cry proteins.
So the temperature in the room is rising.
Right, but this is where the thermostat comes in. Eventually, the room gets too hot. The levels of purr and cry proteins get so high that they start to bind together, and then this purr and cry complex drifts back into the cell's nucleus and binds directly to the clock cycle heater.
And when it binds to the heater?
It shuts it off. It inhibits its own production. This is the crucial negative feedback loop.
Okay, so the heater's off, no more purr and cry are being made. What happens then?
Now the room starts to cool down. Over the next several hours, the existing purr and cry proteins naturally degrade and break down. The heat dissipates.
And once enough of them are gone,
The inhibition is lifted. The purr and cry complex falls off the clock cycle heater. The thermostat clicks back on, and the whole process starts over again.
and that entire loop heat up, trip the thermostat, cool down, restart, takes about 24 hours to complete.
Almost exactly 24 hours. And this elegant chemical timer is ticking away in every single cell of your SCN, all synchronized by the morning light, to drive the rhythm of your entire life.
This is amazing. So that handles the when we sleep. But what about the active sleep itself? We tend to think of it as just turning the brain off, but the book makes it very clear sleep is an incredibly active process.
It's not one state. It's a series of distinct brain states that are actively generated and controlled by at least four different interacting neural systems.
Okay, let's run through the four key players as laid out in Figure 14.28.
First, you have a system in the forebrain, specifically the basal forebrain. This system's job is to generate slow-wave sleep, the deep, restorative stage of sleep. If you electrically stimulate this area in an animal, it will immediately become sleepy.
So that's the go to sleep button.
Pretty much. Second, you have a system in the brainstem, the reticular formation. This is the opposite. This is the wake-up system. It projects axons up throughout the entire brain and wakes it up. If this area is damaged, an animal can enter a persistent coma.
Okay, so we have a sleep button and a wake button. What's the third system?
The third is in the pons, another part of the brainstem. A specific area called the subcoeruleus is responsible for triggering REM sleep, the dreaming stage. But it also does something else that is absolutely vital. It causes atonia.
Muscle paralysis.
Exactly. It sends powerful inhibitory signals down the spinal cord that disconnect your brain's motor commands from your actual muscles. This ensures that while your brain is, say, dreaming that you're running a marathon, your legs stay perfectly still in bed.
So you don't act out your dreams.
Right. And the fourth player,
The hypothalamus.
the hypothalamus, acts as the master coordinator, the switchboard operator. It contains a very specific set of neurons that release a neurotransmitter called hypocretin, which is also known as orexin.
Okay, hypocretin or orexin, what's its job?
Think of the hypocretin system as the finger that holds a toggle switch firmly in the on position during the day. It stabilizes the awake state and prevents the other sleep systems from intruding when they're not supposed to.
And when that finger on the switch slips, that's when we get a disorder like narcolepsy.
That is precisely it. People with narcolepsy have a massive die-off of these hypocretin neurons. They've lost something like 90% of them. So the switch between waking, SWS, and REM becomes loose and unstable.
And this leads to the symptom of cataplexy. This is the one that really, really freaks me out.
It is one of the most bizarre symptoms in all of neurology. Cataplexy is when that REM sleep muscle paralysis, the Tony we just talked about, gets triggered while you are fully conscious and awake.
So your mind is on, but your entire body just goes limp.
Yes. And it's often triggered by strong emotion, especially positive emotion like laughter. A person with narcolepsy might hear a funny joke, start to laugh, and then suddenly their knees buckle and they collapse to the floor. They are conscious, they can hear and see everything, but they cannot move a muscle for a few seconds or minutes.
Their brain just got its wires crossed. It
Yeah.
said, oh, big emotion. That usually happens in dreams. We must be dreaming quick. Cut the power to the legs.
It's a terrifying illustration of how fragile the boundary between our states of consciousness really is.
Which brings us to the final and I think most provocative concept from this whole section, the idea of local sleep.
This is a total paradigm shift, and it's covered in Figure 14.35. We used to think of sleep as a global, whole brain phenomenon. Either the whole brain is awake or the whole brain is asleep.
But the research is showing that's not actually true.
Not at all. Using very fine-grained recording techniques, scientists can now measure the activity of individual neurons in the cortex of an awake, behaving animal. And they've found that while a rat is awake and running around, small patches of cortical neurons might briefly go offline.
They just take a quick nap.
Essentially, yes. They enter a sleep-like state with slow-wave activity for just a split second or a few seconds, while all their neighbors are still firing away normally.
So as I'm sitting here talking to you, it's possible that a small cluster of neurons in my parietal lobe could just be asleep right now.
It's entirely possible. And the text suggests this might be the neural basis for performance errors when we're tired. You know that feeling when you're sleep deprived and you just space out for a second, or you're reading a page and realize you haven't processed the last 3 sentences?
Yeah.
It might be that a critical group of neurons needed for that task just went into a local sleep mode for a moment, even while the rest of you was technically awake.
This is really unsettling. It completely blurs the line of what it means to be conscious or awake.
It suggests that awake isn't a simple binary switch. It's more of a statistical average. You are awake when most of your neurons are firing in an awake pattern, but you are almost never 100% online.
So let's try to bring all three of these chapters together. We started this deep dive with Bella and Benjamin, and the stunning discovery that our gender identity seems to be written into the hardware of the brain, largely by hormones we didn't choose.
Then we moved to homeostasis and saw the incredible redundancy and the evolutionary bias of the systems that control our hunger and thirst, often manipulating our behavior to keep us exactly where our body wants us to be, sometimes against our conscious will.
And finally, we've just looked at the clocks. The beautiful, intricate molecular loops that time our lives and the flickering nature of sleep, where even our own neurons might be taking a break when we think we're fully paying attention.
If there's one single takeaway from all of this, for me, it's a sense of humility. We're not the sole rational authors of our own experience that we like to think we are.
We're more like the users of a very complex, very old, and very opinionated biological software suite.
A software suite that runs almost entirely in the background. You don't consciously decide to release ghrelin when your stomach is empty. You don't choose to find a peacock's tail attractive. You don't tell your SEN cells when to produce purr and cry proteins.
These things happen to us.
They happen to us, and they create the fundamental platform upon which we then live our conscious lives.
I really want to leave our listener with that final thought about local sleep. It challenges the very idea of a unified I.
Go on.
Well, if parts of your brain can be asleep while you are awake, are you ever truly 100% present? Or is your consciousness more like a flickering spotlight, moving around a vast, dark stage where different actors are constantly falling asleep and waking up?
And if the spotlight is moving,
Yeah.
who or what is moving it?
Okay, let's not break my brain right before we sign off.
Chuckles.
Ah.
Fair enough. A question for another day.
A huge thank you to the Last Minute Lecture team for pulling all these sources and this amazing text together. This was a heavy one, but I really feel like I understand the machine a little bit better now.
It always helps to know how the plumbing and the wiring works.
Absolutely. And if you want to actually see some of the diagrams we talked about, the Masters and Johnson charts, that beautiful molecular loop in the SCN, or the visuals of the obesity surgeries, be sure to check them out in the show notes.
They're definitely worth a look to make these concepts click.
Until next time, stay curious.
And try to get some sleep.
Mm.
All of you.
Mm.
At the same time.